Hi, my name is Chloe Blunk, and this is week 3 for bacterial staining for Los Angeles Pacific University. Today is Sunday, July 22nd, and I just wanted to discuss the different exercises that I had to do today. The first exercise was going over a simple staining technique where I had to swab the inside of my teeth and gums and prepare a slide to view under a microscope. During the process of swabbing inside and placing it onto the slide, I had to use heat from the flame to help preserve the cells so they wouldn't get damaged or slide off. After that was done, I let it cool down. I had to drop the red Congo stain, the crystal violet stain, and waited for a minute, poured water over it in the staining lid to clear it off, and once that dried, I viewed it under the microscope. And for the first one, that was the direct stain on the slide, I immediately saw bacteria that were circular in pretty good clumps. I felt that was a cocci cells right there. It was pretty visible to see. And the second one, that was an indirect simple stain where I had to swab the inside of my mouth, and before transferring the swab from inside of my mouth onto the slide, I had to first drop the Congo red stain first and then put the swab on and aggravate it. I felt that that was a little hard to do as far as the technique wise, because the red Congo stain ended up soaking into the cotton swab. So when I had to take the clean slide and spread it, when I had to slide it across, it didn't work, so I had to drop another red Congo stain and continue that. So my results weren't very visible for that slide. So and I'm sure that just had to do with that technique right then and there. But I could see some cells, like piece by piece, just by moving the slide around to kind of get a good view of what was on it. And that was just going over simple staining with just one stain process, and that's just meant for coloring the background to see the size and the shape of the cells. Whereas the second exercise had to do with the differential, where it had multiple staining processes, and it was my favorite. But when I prepared each slide with the broths and cultures that I created last week with the E. coli, yeast, and the epidermidis, it was just a very long process to use the first stain of the crystal violet, wait a minute, and then the second one, third, fourth. So that probably took about 15-20 minutes of my time to go ahead and prepare those slides, and then it took about 15 minutes for them to completely air dry without using the heat. To go ahead and prepare those slides, because that can damage for the gram-negative bacteria. And that was a purpose, was to see for the cells if does it absorb, does a decolorization get rid of the color, and if does, then it's a gram-negative bacteria, and if the color remains, then it's gram-positive. So overall, it was pretty simple. Lasted about an hour and a half, 2 hours most. Wasn't a very long process. So it was just interesting to kind of see what was, as far as the first exercise, to see the bacteria that's inside your mouth and see it under a microscope. And for the second exercise with the broths that I had last week, it was really cool to see the same structures, the same observation that I had made last week as far as this shape. They were the same, so that was pretty cool to see. So that wraps up week 3's lab, and I'm looking forward to the next one.